ஹாய் காய்ஸ், வெல்கம் டு லேர்ன் வித் கோக்ஸ். இன்றைக்கி வீடியோக்குள்ளே போகிறதுக்கு முன்னாடி ஒரு ஆட் பார்த்துடலாம். BuyMote ஷாப்பிங் ஆப். இது ஒரு ஆன்லைன் ஷாப்பிங் ஆப். இதில் க்ராசரிஸ் எல்லாம் அவைலபிளாக இருக்குது. மற்ற ஆப்ஸோடு கம்பேர் பண்ணும்போது இதில் குவாலிட்டியும் அதிகமாக இருக்கும், அதே மாதிரி டிஸ்கவுண்ட்டும் கிடைக்கும். ஸோ ப்ளே ஸ்டோர் அண்ட் ஆப் ஸ்டோர் ரெண்டுலேயுமே இது அவைலபிளாக இருக்குது. டிஸ்கிரிப்ஷனில் லிங்க்கும் டீட்டெயில்ஸும் இருக்குது. செக் பண்ணிவிட்டு இன்ஸ்டால் பண்ணி பை பண்ணிக்கோங்க. இது ஒரு ரொம்ப யூஸ்ஃபுல்லான ஒரு ஆப். அண்ட் இதில் ஒரு இம்பார்ட்டண்ட் விஷயம் என்னென்னா டோர் ஸ்டெப் டெலிவரியும் அவைலபிளாக இருக்குது. ஸோ மிஸ் பண்ணிடாதீங்க, யூஸ் பண்ணிக்கோங்க. ஸோ டுடே we are going to see about RFLP, that is restriction fragment length polymorphism. This is a very advanced molecular biology technique. So the aim of this technique is to analyze DNA to detect genetic variation and identify the genes. Here we have to detect the genetic variation in the genes. Okay, then the principle is RFLP. It is a technique that uses restriction enzymes to cut DNA into fragments of different lengths. Okay, then these fragments can be separated and analyzed to identify the genetic variations. Okay, example for this RFLP is majorly paternity in the criminal investigation. This RFLP is majorly used. Okay, then the materials required. Here I will say the kit based procedures. Okay, 3 reference sample, that is 3 or 4 standard DNA will be taken, and one test sample, that is one test DNA is also taken. DNA ladder is standard, that is already given in that kit, and restriction enzymes are used in this procedure to cut the DNA, and 10x assay buffer also used here, and molecular biology grade water. These are the materials required of RFLP. Then the procedure. Here we have to take, I told you already, there are 3 or 4 standard genomic DNAs are there. That in that DNA sample 1 is taken in the first Eppendorf tube. 50 ml DNA sample 1 is taken. Then 10x assay buffer is 3 ml we have to taken, and molecular biology grade water is 10 ml. And restriction enzyme, here 2 restriction enzymes are used, that is E. coli and PST1. Okay, these 2 enzymes are 1 ml we have to add each. So total 30 ml in each tube, each Eppendorf tube we will have 30 ml of sample. Okay, this is for first genomic DNA sample, that is standard sample. So we have to repeat this for other 2 or 3 standard DNAs. Okay, DNA sample 2 50 ml, the quantity is always same, but the samples will be differ here. In first tube we have to add standard 1 sample 1, here DNA sample 2, and next tube DNA sample 3, and we have 4 means DNA sample 4 standard sample we have to add, and in fifth well we have to add the test sample. Here we only have 3 standard samples, so I have test DNA sample in the fourth Eppendorf tube. That remaining procedures will be same. Then after using micropipette, we have to gentle mix all the sample. We have to change the micropipette from chemicals to other chemical. Okay, and incubate in PCR for 2 to 3 hours. This incubation is very important. In this time duration we have to prepare agarose gel to run the electrophoresis. Then we have to mix the sample using dye, because this dye will detect the movement of DNA in the agarose gel. So definitely we have to add dye in each tubes. Okay, then we have to run agarose gel electrophoresis for 25 minutes. These agarose gel electrophoresis, some steps here. We have to take 2% agarose, that means 2 gram agarose in 100 ml TAE buffer. We do not dissolve in distilled water, we have to dissolve in TAE buffer, that will be 1x concentration. Then we have to add ethidium bromide, that is carcinogenic, so we have to handle it very proper PPE, okay, that must be 0.5 mul. Then we have to add 2 mul dye in PCR sample, that means in that each tube we have 4 or 5 tubes, sample tubes. In each tube we have to add that sample dye 2 mul, okay. Then 15 mul PCR sample is already there, 2 mul, and we have to add DNA ladder in the well of the agarose gel. We have in first well we have to add DNA ladder, and the second well the standard sample 1 DNA sample 1, and next well 2, and next well 3, and next well 4, and last well we have to add the test sample. By this we will check the standardized DNA samples to the test sample. We will detect or conclude the result of movement by checking with the markers, that means the standard DNAs, okay. I told you earlier the standard DNAs genome, that is this: E. coli K12, E. coli 0103, Bacillus cereus, and Bacillus subtilis, and plus one test DNA. These are the procedures we have to carry out to follow the RFLP. So thank you for watching. Follow for more videos. Thank you.